happy to be here to present Herantis Pharma. We developed disease-modifying therapy for Parkinson's disease. As you know, there is a great unmet clinical need there. We are a public company, so the forward-looking statements listed at Helsinki First North. So our lead compound, we call it as HER-096. It's a peptidomimetic molecule that actually mimics the active site of a unconventional neurotrophic factor called CDNF. The HER-096 program is currently in phase 1b clinical study, first time in Parkinson's patients. Last year we completed phase 1a clinical study demonstrating good safety profile, but very nicely efficient brain penetration as the route of administration is subcutaneous. What makes us maybe a little bit different or unique is that we actually have clinical data with the same mechanism of action. So we actually took the CDNF protein through preclinical studies into patients. We treated 17 fairly advanced Parkinson's patients with intracranial administration of the protein, CDNF protein. And among those 17 patients, we were able to show several clinical benefits or improvements and also biological improvements. And now we are using the same mechanism of action, but now we can use subcutaneous administration. As with the protein, we had to use intracranial administration. So basically, during the phase 1 study with the protein, we already understood that intracranial administration is not something that we can commercialize. So we identified the active site of the protein. We developed a D-peptide that mimics the three-dimensional structure. So we have a fully synthetic molecule. And now, of course, we have all the benefits with the synthetic molecule there. And what we address as a mechanism of action is a pathway called unfolded protein response pathway within the neurons. So what we are looking at is actually, of course, long-term disease modification effect, but we also have seen very robustly quite rapid symptomatic improvement. And we are looking at subcutaneous administration 1 to 2 times or 1 to 3 times per week in the longer run. You might know that Parkinson's disease really is urgently needing new innovation. Currently, the patients are treated with symptomatic treatment, but however, the current treatment is only effective in maybe 2/3 of the patients, and nothing can slow down the progression of the neuronal damage in the midbrain. So there are 10 million people in the world who are suffering from this. And how we are actually addressing this. So no one knows what triggers Parkinson's disease. However, we know that the neurons in the substantia nigra are suffering from a vicious cycle composed of endoplastic reticulum stress, accumulation of toxic alpha-synuclein aggregates, and neuroinflammation. And each of these components strengthen each other. And all of this results in decreased dopamine secretion and thereby movement disorder. However, we can, in a way, rescue or regenerate the function of these degenerating neurons. And we address the unfolded protein response pathway within the ER. We have shown robustly that we can decrease the amount of alpha-synuclein aggregates, we can alleviate the neuroinflammation, we can increase the dopamine secretion, and we can normalize the motor function in preclinical settings. So let's have a look on the data. We have used an animal model that combines aging, alpha-synuclein aggregates, and progressive disease. And of course, the first question is, can we modify the target pathway by subcutaneous administration within the brain? And we have used 2 UPR markers here. In the graphs, the white bars represent the healthy aged animals. The red ones are the model animals, and the blue ones are then treated animals. And as you can see with the red bar, we have a constant activation of the target pathway, which we can nicely bring down the activity with subcutaneous HER96. Nice thing. How about disease modification? A hallmark in the pathology of Parkinson's disease is alpha-synuclein aggregates. We have an alpha-synuclein aggregate model, so we have a high concentration of those in the brain of these animals. We can bring that nicely down by 50%. Neuroinflammation, we can actually normalize completely with a 4-week treatment of these mice. Nice. But how about the effect on the dopamine system? The first graph here shows the protection of dopamine neurons. We have up to 60% more dopamine neurons with the treated animals, which coincides in completely restoration of dopamine levels in striatum and normalized motor function, which is maybe best visualized using this video. So the first mouse here is a just normal aged mouse walking with a bar walking test for motor coordination, no problems there. The second one has the induced pathology treated with placebo. As you can see, we have a very clear motor phenotype within this model. This guy is really struggling there. And then we have a mouse here, which has the pathology treated for 4 weeks with HER96 subcutaneously, and wow, we can go into Olympics with these mice. So as you can see here, at the same time, running the treated animal is actually quicker than the normal mouse. So, of course, extremely happy about this. As I said, we have completed phase 1A clinical study last year, a classical single ascending dose study in healthy volunteers, placebo-controlled from 10 to 300 milligram doses. Great safety profile, no problems there. Then we had a secondary cohort, elderly, healthy individuals for CSF collection to measure the blood-brain barrier penetration. And now in this graph, each of the data points represent one individual from which we took the CSF sample at a certain time point. And as you can see, we end up in a concentration of 50 to 100 nanograms per ml in CSF, which is exactly within the therapeutic range based on the preclinical studies. So extremely happy about this very successful study. And now we are currently running the phase 1B clinical study. Actually, 2 weeks ago, we announced that we already completed the first part, which completes the PK part or PK studies in CSF. And now we are moving into patient treatments. We recruit 24 early to mid-stage Parkinson's patients who will be treated for 4 weeks, 2 times a week. Of course, safety is the primary endpoint, but then we have very much ambitious biomarker program in order to identify biomarkers that can demonstrate the target pathway modulation in patients. This is also very important towards phase 2. We also have a wearable device for continuous monitoring of the motor symptoms. So we have great data on neurorestoration. We have patient-friendly administration route, and so far, extremely good safety profile. Some words about the company financing. Extremely happy that the phase 1B study is fully financed by a consortium of Michael J. Fox Foundation and Parkinson's UK. Together with European Innovation Council financing, we have raised more than 6 million euros non-dilutive financing during the past 18 months. And we also have a commitment from EIC Fund for equity investments. They can chip in 1/3 of anything that we raise in the future. So basically, what we are currently doing, we are preparing for phase 2 clinical studies. The phase 1B is fully financed. We are, as a public company, contemplating about a capital injection to boost up the phase 2 preparations. One final word is that, are we a one-trick pony? No, we are not. The target mechanism is very relevant for different neurodegenerative diseases and also other diseases, for example, metabolic diseases. So happy to have questions and maybe discussion after my presentation. Thank you.
Thank you so much. You said you're not a one-trick pony, so then I think this question is quite relevant. Have you tested your product also as a preventive compound?
Yeah, good question. The challenge is that when you diagnose Parkinson's disease, it's always based on clinical findings. And at that stage, when you have the first early symptoms, the disease has actually progressed pretty much. So typically 50% of the activity of the certain area, neuron activity of the certain area is already lost. So basically we cannot prevent because there is no diagnostics for that. But hopefully we can stop the progression at that stage when the symptoms are very, very mild and so far in such keep the good quality of life in a way thereafter.
Do you have effect on NFL?
Neurofilaments, I guess.
Thank you. I was just like, yeah, I hope so. I don't know.
That's something that I don't know. We haven't investigated that, so no clear answer for that. I don't know.
You don't know, exactly. You talked a little bit about the finances just in the end, but if you missed out, what is your financial status, say, by the end of the year?
Yeah, so basically we have the finances to run the phase 1b clinical study, expected to have readout in about a year. But then, of course, we want to be prepared for phase 2, so requiring maybe some additional investments. And of course, the objective is that once we have the phase 1b readout, we can discuss with the pharma partners that this is the data package and this is the phase 2 plan. Are you interested in joining? As we have pretty advanced discussions with many pharma companies already.
And I have a question about the video, of course, maybe the highlight of the presentations with the little mice. Have you repeated the bar walk in a second independent lab, and do you have a relevant sample size?
Yes, we do have relevant sample size. We have actually used just one laboratory in France because they have this model, they have developed the model. And yes, we have repeated the experiment.
Is your in vivo model validated and the same as used for the other compounds?
That's a tricky question. Yes, our development partner there is a French CRO, and they have, yes, validated that. I'm not aware of all the studies that have been done with that model. Of course, that's confidential information of the CRO and then the companies using that.
Here comes a moral question. One of your core values as a company include integrity. Can you tell me about a time when it was hard to speak up or take accountability, let's say, or how do you define integrity in this sense?
That's a surprising question.
Yeah.
I'm not used to that.
Well, maybe they didn't look at your homepage.
Yeah, exactly.
Well, but I did.
Yeah. No. It really means that we try to be really honest and really straight about what we have and what we don't have.
Do you think that's lacking in this business, this kind of frontness?
Maybe not within Nordic companies, but sometimes when you go a bit further, it's possible.
That's a nice and diplomatic answer, but I think we all got the context of it. I don't see any other questions in the room. Anything you would like to add?
No,
I think that answered everything. Thank you so much for your presentation.
Thank you. Thank you.